Good morning, students. Today, let us study about one more important fungal pathogen called as Cercospora. Cercospora. This is one of an organism which recently it has been placed under the subdivision Deuteromycotina. I will elaborate why now it has been placed under Deuteromycotina, and this particular organism also has anamorphs in ascomycetes, that is the perfect stage of Cercospora has been placed under the subdivision Ascomycotina, whereas the asexual reproductive part of this particular species is called as Cercospora, where I'm going to elaborate about this in my next slide. So however, today we are going to study about Cercospora species which cause an important disease in case of groundnuts. The disease is commonly called as Tikka disease. It is also called as leaf spot disease. So let us study this in detail. So as I was mentioning, Cercospora species which cause a disease called as Tikka disease. It's also called as what? Leaf spot disease. Why it is called as leaf spot? Because on the surface of the infected leaves of these groundnuts, you can find such kind of necrosis, that is blackish or brownish spots can be observed on the surface of the leaves. Based on the symptom, this particular disease is also named as leaf spot disease. The pathogen is Cercospora, which infects groundnuts. So the botanical name of these groundnuts are Arachis hypogaea. You all know about that. So you know these groundnuts are highly economically important, isn't it? This groundnuts, they are the one which are oil yielding crops. If the destruction of this crop has happened, this will be a great economical loss. Hence, this particular disease has to be studied, and also we have to study how to manage this particular disease or how to control this particular disease, and which is the pathogen which causes this disease, which is the climatic condition required for the spread of this particular disease, and the disease cycle of this particular organism. Let us study in detail in this particular lecture. So as I had mentioned, Cercospora species, that is the imperfect stage, that is asexual. There are species of Cercospora where, where you can find only the asexual reproductive phase. Sexual reproduction is completely lacking. Such group of Cercospora, they have been named, mycologists named them as Cercospora, and it has been placed under the subdivision Deuteromycotina. Students, you all know what is the significance of this subdivision Deuteromycotina? The organism where the sexual reproduction is completely lacking. Such group of organism has been placed under the subdivision Deuteromycotina. Hence, the Cercospora, because of the lack of the perfect stage in the sense what students? The sexual reproductive phase is completely lacking. Hence, the mycologists have placed them under Deuteromycotina. There are few species of Cercospora where they are actually the anamorph, that is the another morphology of similar morphology of Cercospora, where this organism has an ability of undergoing the process of sexual reproduction, and they produces a spore called as ascospore. Hence, they have been placed that under Ascomycotina. Hence, students kindly remember the anamorph phase or the anamorph of Cercospora is Mycospirilla. This is one of the important question for the point of your competitive examination. Anamorph of Cercospora is Mycospirilla. Why this Mycospirilla has been placed under Ascomycotina? Where during the life cycle of this particular organism, even though the morphological feature of this Mycospirilla is same as that of a Cercospora, but The only difference between the Cercospora and Mycospirilla is Cercospora undergoes the process of only asexual reproduction, which is the characteristic feature of Deuteromycotina, whereas Mycospirilla, which resembles to that of a Cercospora in its morphology or in genetics or in the process of pathogenicity, everything is the same, but they have an ability of undergoing a say sexual reproduction, where the product of the sexual reproduction are ascospores. Hence, the sexual reproductive phase of Cercospora is called as anamorph, and the genus is called as Mycospirilla. So this Cercospora has been placed under the division Eumycota because they are true fungi, subdivision Deuteromycotina because they are imperfect fungi. Imperfect fungi in the sense they lack perfect stage. Perfect stage in the sense sexual reproduction is completely lacking, and they belong to the class Hypomycetes, order Moniliaceae, family Dematiaceae, and the genus of today's class, which we are going to study, is Cercospora. So, this is one of an organism, as I was mentioning, it causes a disease called as leaf spot, which is commonly called as Tikka disease. There are two different kinds of leaf spot which you can come across in groundnuts. There are two in a single host, groundnuts are the host for Cercospora. In the single host, you can come across two different kinds of spots which are caused by two different species of Cercospora. Which are those species? Which are the two different kinds of spots you can come across in groundnuts? One is early leaf spot, that is the spot or the symptom which you can find early after the 2 weeks of the planting this particular plant or the seed which we have sown. After about 2 to 3 weeks of the germination of this particular seed, the infection start. The first point of infection is by the Cercospora arachidicola. Hence, it is called as early leaf spot. Hence, it is called as early leaf spot. And later on, due to the secondary infection, the primary infection is usually through Cercospora arachidicola, and the infection start at the earliest. Hence, it is early leaf spot, and also the symptoms of early leaf spot and the late leaf spot is different. I am going to tell you that later also. And another kind of spot which you can come across in the same host, it is called as late leaf spot, where the formation or the visible symptom which you can find on the infected plant is little late than that of early spot. And the species which infect and cause the late leaf spot is Cercospora personata. Once again, I will repeat. You can come across two different kinds of symptom or two different kinds of spot, leaf spot on groundnuts. One is early leaf spot, which is caused by Cercospora. Remember students, this is very important for the point of your competitive examination. Early leaf spot disease caused by Cercospora arachidicola. The anamorph is Mycospirilla arachidicola. Late leaf spot, that is caused by Cercospora personata. Remember students, Cercospora personata, which causes late leaf spot. And this particular disease is caused by Cercospora personata. Anamorph is Mycospirilla burkilgii. These are the two different species of Cercospora which causes Tikka disease or leaf spot in case of Arachis hypogaea. So now we studied about the pathogen, and also we studied about the systematic position of this particular pathogen, and also we studied the different kinds of spot it causes and the different species of Cercospora which causes early leaf spot and late leaf spot. Now, coming to the distribution, students, you have to write in the same order in your examination. In your view examination, it has to be written in the same order. Distribution. This particular disease is worldwide distributed. Wherever the groundnuts the groundnut crop has been grown all over the countries, including US, China, India, wherever this particular groundnuts have been grown. It is epidemic wherever it has been grown. The infection can be observed worldwide. And due to the infection of this particular disease, the yield of the crop decreases. Approximately around 22% of the yield will be decreased because of the infection of this particular pathogen. Now, coming to the difference between, as I was mentioning, students, before also, I was mentioning that there are two different species of Cercospora which causes the leaf spot disease. Which are those? One is Cercospora arachidicola, Cercospora personata. These are the two different species which causes spot disease, leaf spot disease in groundnuts. What is the difference between these two pathogens? Cercospora arachidicola causes, as I had mentioned, early leaf spot. It causes early leaf spot. Whereas Cercospora personata causes late leaf spot, that is early leaf spot. Why it is called as early leaf spot? Because the visible of this particular symptom is earlier than that of the visible symptom which is being caused by Cercospora personata. Then coming to the mycelium. What is mycelium, students? This is the thread-like structure or the vegetative body of the fungi is called as mycelium or hyphae. Mycelium, how these mycelium of Cercospora arachidicola? It is intracellular, whereas Cercospora personata, it is intercellular. What is the meaning of intracellular and intercellular? Intracellular, students kindly note down intracellular, that is the pathogen, that is the mycelium, this kind of mycelium which would have penetrated inside the host tissue, they are found to be intracellular, means they can even enter into the host cell. They can enter into the host cell. Because they are entering inside the host cell, they do not require any hostorium for the process of absorption. They do not require any hostorium for the process of absorption. Whereas Cercospora personata, it is intercellular, means in between the cells of the host, you can find this particular mycelium in the infected plant, and Cercospora personata has an ability of producing branched hostorium. What are these hostorium? These are some projections which are produced. These are the modification of the mycelium or the hyphae of the fungi, where this projection can enter into the host cell, helps in, the main function is to absorb the nutrition from the cell. So the Cercospora arachidicola, it is intracellular. What is the meaning of intracellular? They can enter inside the host cell. Because it is entering inside the host cell, they need not require hostorium for the process of absorption. Whereas Cercospora personata, it is intercellular. That is, it is found in between the cells. Because it is found between the cell, they require certain hostorium for the process of absorption. So this is the difference between the mycelium of arachidicola and personata. And if you see the color inside the parasite, inside the host, it is brownish in color, whereas it is blackish brown in color. Then coming to the conidiophore, what is their conidiophores? Wherever I'm pointing it out, so this is the infected leaves. Infected leaves, if you take a transverse section, you can see the hyphae entering inside the host tissue. It can be intercellular or intracellular. And they enter into the reproductive phase. During the process of reproduction, what they does, like in rust disease and all, they also start producing the spores. So how they will produce the spore? They produce the spore by producing a structure called as conidiophore. So these structures are called as conidiophores. They produces reproductive hyphae called as conidiophores. The conidiophores at its terminal region has an ability of producing a sexual spore called as conidium or conidia. Okay, so here the conidiophore of Cercospora arachidicola, it is unbranched. both personata as well as arachidicola, both the conidiophore. Once again, what are these conidiophores? These are the structures which bears or which produces conidia. They are called as conidiophore. So these conidiophore, these conidiophores are unbranched. They are unbranched, and and it is for in case of arachidicola, the sporidiophore is around 22 to 44 microns in length and about 3 to 4 microns in width. Whereas personata, it is 25 to 54 microns long. Comparatively, they are longer than that of arachidicola, and it is 5 to 8 microns in its width. That is the difference between the arachidicola and personata. And the conidia, coming to the conidia, the spores of or the conidia of arachidicola is comparatively longer. See this, comparatively longer than that of personata. See, if you see this, this is shorter and these are longer conidiophore. So here in case of arachidicola, the conidia is long and cylindrical, pale yellow in color, and it is 38 to 108 microns and 6 to 8 microns in width. Whereas the personata, the conidia is comparatively short. Imagine, they are comparatively short, cylindrical, and the length of this particular conidia is comparatively shorter than that of arachidicola. And the shape, if you see the shape of this conidia, in case of personata, in case of personata, it is round at the base and tapering towards another end. That is the characteristic feature of personata. And they have got a cross walls that is called as septa. The septa is around 3 to 4 septa you can come across in case of personata. In case of arachidicola, as I was mentioning, the conidia is comparatively very long and it is truncated. What is the meaning of truncated? Truncated means just chopped out. See, for example, tender coconuts and all, they will chop and give, no? In the say that is called as truncated. Such kind of cut or chopping like structures you can come across. That's called as truncated. And also, they are having 1 to 12 septa can be observed. So this indicates that the spore or the conidia of arachidicola is long and has got many septa, and whereas personata, the conidia is completely shorter and it has got a round base and tapering ends, whereas in case of arachidicola, it is truncated. So this is the difference between the conidia of arachidicola and personata. And if you see the mode of nutrition, both these two organisms, they are parasitic and saprophytic. So they can grow as a parasite and cause disease, and even they can survive even as a saprophyte. That is, this indicates that they are facultative parasites. That is, it may or may not require host for their growth and development. Once again, I will repeat. There are two different species of Cercospora which cause such kind of leaf spot disease in case of groundnut. One is Cercospora arachidicola, Cercospora personata. Cercospora arachidicola causes early leaf spot, whereas personata causes late leaf spot. Arachidicola is intracellular, whereas personata is intercellular. Arachidicola lack hostorium, however, this personata, they have hostorium. Conidiophore, both they are unbranched, and conidia in case of arachidicola is longer than that of Cercospora personata, and both are facultative parasite. This is about the pathogen. This is about the pathogen and the difference between the Cercospora arachidicola and Cercospora personata. Now, coming to the symptoms of this particular disease. Symptoms means what? Visible changes on the surface of the host. That is called as symptoms. What are the symptoms we can come across? Which are the different symptoms we can come across in this groundnuts if it has been infected by Cercospora? As I had mentioned, there are two different spots which you can come across in groundnuts. One is early, other one is late leaf spot. So when this early leaf spot formation happen, it is it it happen before that of a late leaf spot. That is, by about 2 to 3 weeks of the germination of the seed, you can come across this particular leaf spot. Whereas this also because of the secondary infection. It happens in the later stages, and the infection, that is early leaf spot, is due to the early infection or due to primary infection, whereas late leaf spot, where the infection happens late and also it is due to secondary infection. And where this infection happen, upper surface of the leaf, both you can see on the upper surface of the leaf. And how the spots, you can see these spots early, and this is the late leaf spot. How actually you can see the spot? Arranged random, wherever the infection has happened, it is random, whereas here it is concentric rings. What is the shape of the spot? It is circular to irregular. The spot of early leaf spot is circular to irregular, whereas in case of late leaf spot, it is always circular. And another important thing which we have to note down over here is the spot which is being caused by Cercospora arachidicola, that is early leaf spot. You can find this yellow hollow can be, this is the chlorosis, and this black colored is the necrotic spot. We can see this predominant yellow hollows can be seen on, we can see it on the infected leaf. See, if you see this leaf and this leaf, you can see brownish spot, whereas these spots are blackish in color. Remember, the arachidicola, the spots are quite irregular, and they produces brownish spot, and it is being surrounded by yellow hollow. That is the hallmark of this particular disease. However, in case of late leaf spot caused by Cercospora personata, it is the spot is charcoal black in color. May or may not, you can see the yellow hollow on the surface, on the outer surface of this particular spot. That is the characteristic feature. And another important thing which we can see here is at the lower surface, you can see the infection of the Cercospora arachidicola even at the lower surface. Same leaf, if you just turn and see, you can see the spot, and also you can see the formation of the conidiophore at the lower surface of the leaf. That is very much important to differentiate Cercospora and arachidicola. See, if early leaf spot, the infection only at the upper surface. In case of late leaf spot, the infection, that is the symptom, black spot can be seen on the upper surface, prominent spot can be seen. If you just reverse that and see, you can see comparatively smaller spot which consists of conidia at this particular region. And this is black in color, whereas the early leaf spot, they are brownish in color and surrounded by yellow hollows. And also the size, spot size, the early leaf spot, the comparative larger than that of a late leaf spot. So these are some of the disease symptom, and also you can see this, as the disease symptom, the name of the disease tell, spot disease, leaf spot disease. So the predominantly the infection happened to the aerial part of the plant. However, sometimes even the infection can be seen even onto the groundnuts. Where you can see the spot even on the shell of this groundnuts, because you know groundnuts, after the process of fertilization of the flowers, the inflorescence just bend down. If you would have seen the groundnuts, they are actually what? They are actually formed from flowers. They are not actually the root or stem modification like that of your radish, potato and all. You know that. They are actually the seeds which has been formed due to the fertilization of the flower or the ovary. Once the fertilization has happened, you could see that particular flower or the inflorescence just bending down, and the formation of this particular seeds or the fruits happen at the ground level. So by that time, even the flowers also would have got infected. That is the reason you could see this spot even on the shell of this particular groundnuts. So this is about the symptom of this particular disease. So coming to the environmental factors which are required for the spread of the disease. Prolonged high relative humidity. Humidity is the main reason for the spread of this particular disease. Temperature, low temperature, that is the temperature around 20 degrees Celsius with dew on the leaf surface also helps in the spread of the disease. Heavy doses of nitrogen and phosphorus fertilizers. The pathogen survive for long period in the infected leaf debris through conidia, dormant mycelium in the soil. The primary infection is by ascospores or conidia from infected plant debris or infected seeds. Once again, I will repeat, the environmental condition required for the spread of the disease is humidity. Humidity factor play a major role in the spread of the disease. Temperature around 20 degrees Celsius with dew on the leaf spot. So that also causes the infection. And also, if you are using heavy doses of nitrogen and phosphorus, may also lead to the spread of the disease. And if the plant debris is being retained in the soil, proper sanitary measures have been not followed. So in such a case, the infection can happen. Primary infection is by ascospore or by conidia from the infected plant debris or infected seeds. So this is about the environmental condition required for the spread of the disease. And the secondary infection, primary infection is by the plant debris, where the plant debris, if they have some conidia or the dormant mycelium, they can produce the conidia and the infection can happen. The secondary infection happen from the infected plant. If you can find any infected plant producing conidia, these conidia are dispersed through wind or through rain splashes. The conidia gets dispersed and infect the plant. So now let us study how this infection can happen. So let us study about this disease cycle. So this is the cycle of Cercospora arachidicola, and this is the disease cycle of Cercospora personata. As you all know, Cercospora arachidicola causes early blight and Cercospora personata causes late blight. So here, if the plant debris, say for example, any plant debris is left out in the soil, the crop residue, if you can find in that particular field, onset of favorable condition, the mycelium which are present in this crop debris produces ascospores. I am not going to elaborate how these ascospores are produced and all, not required for you all. They produces ascospores. These ascospores serve as a primary infection. I was mentioning the primary infection happen through ascospore or through conidiospore. So if you can find the previous plant debris in the soil, during favorable condition, the mycelium which are present in this plant debris, they produces ascospores. These ascospore causes primary infection. Even these plant debris can also produce conidiophore. They can also produce conidiophore. From these infected lesions, you can see the formation of these conidiospores. These conidiospore can get disseminated through wind or insects or utensils or any other way. They get disseminated. Once it land on the host, it germinate. How actually it germinate? These ascospores or conidiophore produces germ tube. This germ tube enters into the host tissue. This is the process of infection, how it is happening. See, this conidiophore has fallen on the surface of the host, on the leaf, for example. They are germinating and they are producing germ tube. This germ tube can enter through this stomata. They grow and develop inside the host tissue. How it is growing? This is Cercospora arachidicola. They are found to be intracellular. That is, they can enter even into the host cell. They can enter into the host cell. They are not producing hostorium. They grow, develop, and they undergo a process of reproduction. Another important process thing you have to remember here is the Cercospora produces one toxin called as cercosporin. Remember, students, They have an ability of producing a toxin called as cercosporin. Because of the production of this cercosporin, there will be a damage in the cell which leads to the death of the cell. That is why you can see such kind of black spots or necrosis. And from the same lesions, you could see the formation of the conidiophore and the conidia from the same lesions. And if this conidiophore get dispersed through wind or through rain or through insects or through utensils or whatever it is, once it come in contact with the healthy leaf of the groundnuts, the infection can happen and the cycle continue. Here also, in case of Cercospora personata, if you can find the debris which has been found in the soil for a longer time, if proper sanitary measures have been not followed, these debris has an ability of producing ascospores. These ascospores causes primary infection, and these ascospores germinate. They germinate, enters into the host through stomata, and they are what? Intercellular, I was mentioning. They are intercellular. What is meaning of intercellular? In between, they cannot enter into the host cell like this. So here the mycelium is entering inside the host cell, whereas here the mycelium is in between the cell, and they are producing some branched structure called as what, students? Hostorium. Through this hostorium, they can absorb the nutrition, and at the same time, even they have an ability of producing the toxin. This toxin leads to the formation of such kind of black spot. And at the lower surface of this particular spot, if you observe this, you have to remember this conidiophore is forming at the upper surface of the leaf, whereas in case of personata, it is forming at the lower surface of the leaf. These conidiophores are produced at the lower surface of the leaf, and these conidia which has been formed, once they have been disseminated through rain or wind or insects or any other mode, once it come in contact with that of a healthy plant, the cycle continues. This is about the life cycle of Cercospora arachidicola and Cercospora personata. Now, how to manage this particular disease? One easy method is always go for crop rotation. So if you are growing groundnuts this season, better to go for some other pulses, or for example, rice, ragi, wheat, jowar, sorghum, such kind of plants can be planted. So crop rotation can be done. Eradication of infected plants by burning. Whichever the plant has been infected, take out, uproot that particular plant and burn into ashes. That is called as eradication. Pruning, again, remove that particular part of the plant using a scissor, remove them and burn it into ashes. And if you are using the seed for the process of sowing, if the seed, there are chances of seed also to get contaminated. So we can sterilize that particular seed by disinfecting it with low concentration or dilute sulfuric acid. Or if you are using only the seed without shell, then you can treat it with 0.5% copper sulfate solution, dip it in copper sulfate solution, so that the Cercospora, if it has got infected, also get killed. That particular process is called as seed treatment. Before sowing the seed, go for seed treatment with copper 0.5% copper sulfate solution. And the seed can also be treated with thiram and spraying of carbendazim, commonly called as Bavistin, 500 grams, or mancozeb, 2 kg per hectares. We can spray that to avoid this disease. Better to go for disease resistant varieties. So we have some of the disease resistant varieties: ALR1, GJG32, TG37A. These are some of the disease resistant varieties where this groundnut plants, if you are using, may not get infected with this particular pathogen at all. So these are the different methods of managing this disease. One is crop rotation, eradication, disinfection, sanitation, treatment with carbendazim, mancozeb. Even Bordex mixture also gives very good results, and you can go for disease resistant varieties. And students, these are some of the references which you can use. And thank you. Students, if you have any question,